Hello friends, today we are going to study about protein folding part 2, and in this video we're going to study about the proteins that helps in folding of other proteins. So the proteins that helps in the folding of other proteins, these proteins are known as molecular chaperones. So what are molecular chaperones? These are proteins only, but these proteins help to fold other proteins. These are mainly heat shock proteins and mostly require ATP for their functions. There are various classes of molecular chaperones. Among these molecular chaperones, the first one is HSP70. The alphabets HSP means heat shock protein. That means these are abundant in the cells that are stressed with high temperature. These HSP70, they bind to the hydrophobic region of unfolded protein and prevents their aggregation. Now, why hydrophobic region? How a cell will know that the protein is properly folded or it is unfolded? Because in case of globular protein, we already studied that the globular protein has two sections. One section is its interior, which is completely separated from the exterior aquatic environment, and the exterior portion, which is in contact with the aquatic environment. So this interior of the globular protein consists of hydrophobic core. So all the hydrophobic amino acids are present in the interior of a globular protein. So if these hydrophobic amino acid is found in a protein where it is present outside, that means that protein is not properly folded or it is unfolded. Okay, so if the protein is in a properly folded state, all its hydrophobic amino acid should be in the interior of the protein. If any hydrophobic amino acid is at the outside region, that means the protein is not properly folded. So these HSP70 also recognize the protein by seeing its hydrophobic region. If it is exposed outside, that means the protein is not folded. So it will bind to that hydrophobic region and prevent their aggregation. Now, HSP70 is found in prokaryotes also, in eukaryotes also. In case of eukaryote, it is known as HSP70, and in case of prokaryotes, it is known as DNA-K protein. They helps in the folding of newly synthesized protein with the help of another chaperone, which is known as co-chaperones. So in case of prokaryotic, that helper protein is known as DNA-J, and in case of eukaryotes, this helper protein is known as HSP40. So HSP70 and HSP40, where HSP70 is the main chaperone protein that helps in folding with the help of HSP40. Similarly, in case of prokaryotes, the main protein that folds is DNA-K and takes the help of DNA-J. They can also reverse the denaturation and aggregation of protein, and sometimes they help in unfolding of protein during transport. And once the transport is done, they again fold the protein. So let's understand the mechanism of HSP70 in prokaryotes, that is DNA-K and DNA-J. This is an unfolded protein. What happens? First of all, in this unfolded protein, DNA-J will come and bind. Once DNA-J will come and bind to this unfolded protein, it will direct the binding of DNA-K. DNA-K has ATP attached with it because the process of folding requires energy. So now to this unfolded protein, DNA-J, which is already bound, will help binding of DNA-K. Once the DNA-K bound with this unfolded protein, then it will start its folding, which requires ATP. So therefore, it will use its ATP, and hence what will happen? The ATP will get converted into ADP. The next step is this ADP should again get replaced with ATP so that this DNA-K can start its second round. That means it can help in the folding of other unfolded proteins. So for that, we need a molecule which is known as GRPE. GRPE is a nucleotide exchange factor. That means it will help to exchange ADP with ATP. So now again ATP come and bind with DNA-K. The next step will be the folded protein will release out, and this DNA-K is now free for its next cycle. So this how DNA-K and DNA-J work to fold an unfolded protein. Apart from HSP70, one more molecular chaperone is chaperones. These are found in prokaryotes as well as in eukaryotes. They bind with improperly folded protein and help in their folding. Example is GRO-EL-ES chaperone. Now how it works? Here, this structure is GRO-EL. Now it has two chambers. One is known as cis ring, one is known as trans ring. What happens is the unfolded protein will first enter in the trans ring along with ATP molecules. From here, it gets shifted to cis ring, the unfolded protein as well as the ATP. Next, this GRO-ES, which is a cap, will come and bind, so that now this becomes an enclosed structure. Now inside this enclosed structure in the cis ring, the folding will take place with the use of ATP. The total of 7 ATP is used. So PI will release, ATP will get converted into ADP. Now once the protein is folded, now in the next step, again an unfolded protein along with ATP will come and bind to this trans ring. And after that, the cap will open and the folded protein will release out. Once it is released, again this unfolded protein will shift from trans ring to the cis ring along with ATP. Again, it will come and bind the GRO-ES and the process will repeat. So this is how the cycle continues to help in folding of unfolded protein. Now, apart from HSP70 and chaperones, two other molecular chaperones are. One is trigger factor. Trigger factor, these are found in prokaryotes and these are associated with ribosome. They prevent the aggregation of polypeptide. Trigger factor and HSP70 are the first chaperones that are made in prokaryotic protein encounter. So these are the first chaperones that helps to fold the unfolded protein. Next is HSP90. It is a eukaryotic protein which facilitate the later stage of folding of protein that involves in cellular signaling. So these are the two more molecular chaperones that help in the protein folding. Now, these are some diseases that are caused due to protein misfolding. If any protein remain misfolded, then these are the following diseases that will arise. So first is Alzheimer's disease. The protein whose misfolding will lead to Alzheimer's disease is amyloid beta protein. Next is sclerosis. Protein is superoxide dismutase. Huntington's disease, because of Huntington with polyglutamate expansion. So like this, these are the list of diseases that occur due to misfolding of some important proteins. Now, one question from my end for you all. Not all heat shock proteins are chaperones. Some are proteins that facilitate the degradation rather than refolding of other proteins. Explain why the rate of protein degradation would increase during heat shock. Do let me know in the comment section. If you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.